The topic of present video is stability risk assessment and improvement basics for pharmaceuticals. As the name indicates, stability, risk assessment and improvement. So in this video, we are going to learn about what are the risk factors for stability of the pharmaceuticals, how you will confirm the risk for stability, and how you will improve the stability of the pharmaceutical formulations. Welcome, dear viewers, to this important topic. And after watching this video, I hope you will be able to understand how to do the risk assessment for stability. And you will also learn about the ways to stabilize the pharmaceutical formulations. So let's start with the video. See, first is the risk assessment part, and this risk assessment is for the stability of the pharmaceutical formulation. As you know that the formulation is a composition of the drug, excipients and the packaging materials. It may be the tablet formulation, capsule formulation, oral liquid formulation, suspension, solution or any formulation. It may be injectable or it may be ointment. That means it might be solid, liquid or injectable formulation. So first stage is to do the risk assessment for your formulation. Whether you are working on the new formulation or whether you are working onto the generic type of formulation. How you will do the risk assessment? So first part is API related risk assessment. Then it will come to the excipient related risk assessment. Then manufacturing process related, packaging related, and then the storage related. First is API related. Consider the force degradation data of API. Consider mild degradation data. Sometimes some APIs get much degraded into the force degradation and you will not know exact sensitivity of the API. For those APIs, you can do mild degradation studies and then you can draw some conclusions for the stability risk of the API. Then consider the stress study data, and out of these data or studies you will understand the intrinsic stability and intrinsic sensitivity of the API. Whether your API is sensitive to oxygen, sensitive to moisture, sensitive to light, that information should be gathered. Then what are the possible degradation pathways of the API? Also, many of the scientists perform open exposure study at different RH conditions and at different temperature conditions to know the sensitivity of the API. Then is excipient compatibility and composition related risk. Whether your API requires specific stabilizer or whether it is showing any incompatibility or it is showing degradation with some excipients, that is to be assessed. Formulation design related risk assessment. If you are working on a tablet formulation with break line, then you will need to perform the stability study on the split parts at 25 degrees Celsius and 60% RH in a bottle without seal and without desiccant. So understand these formulation design related requirements early in the development phase so that you will not have any surprise at the later part. Consider you are working on a soft gelatin capsule, so you should do the risk assessment for cross-linking in the gelatin. Then manufacturing process related risk. Whether your API is sensitive to some manufacturing processes or process parameters like temperature, light, or humidity that is required to be assessed. Then storage conditions and duration related risk. Whether the API is having low melting point, whether any excipient is having low melting point, or the composition is showing degradation because of the storage condition or not. Then in use stability, compatibility and reconstitution related. These risks are required to be evaluated thoroughly. Perform the literature search, know the marketed products. If some products of the same API are marketed, know their design and study those products. Also, if you are working on a generic formulation, then understand your reference formulation. Consider all the risk of critical material attributes, critical process parameters, and the packaging related attributes for your formulation. Also, perform the risk assessment of all these CMAs and CPPs for all your drug product sequence. Because the stability is not related only to the impurity or assay or dissolution or other parameters. It includes all the drug product quality attributes, starting from appearance till the water content, impurity, dissolution, if it is a solid oral or liquid oral, and physical appearance, if it is injectable, then pH, particulate matter, microbial growth. So all these parameters are required to be assessed for stability risk. Now coming to the first point, that is excipient related. First point is excipient source and specifications. Excipient interaction and compatibility is required to be assessed. And if you know that the excipient is not compatible with drug, so you can exclude that excipient, or you can change the grade, or you can use other excipient in place of that incompatible excipient. So this is the stability improvement technique for excipient related. Then degradation due to excipient compatibility, incompatibility and impurities. Some of the time some excipients are used into the formulation which contains impurities. Those impurities may interact with the API and that's why it will destabilize the formulation. Also consider API to excipient ratio. Stabilization by excipient quantity, grade, particle size distribution, loss on drying or water content. If your API is sensitive to water or sensitive to moisture, then you can use some excipients which are having low moisture content. Use of basic and acidic excipient to have specific pH, or you can use the buffering agents. You can also use the antioxidant if API shows oxidation effect. Then you can also go for using chelating agents or complexing agents to stabilize the formulation. Use of protective coatings like film coating, moisture barrier coatings are there for tablet formulations. Use of specific capsule shells for capsule formulation. Use of solvents, use of stabilizers, water or opacifiers. Opacifiers or formulation coating containing opacifier is used to protect from light. Use of special grade excipient like low nitrite, low peroxide or low loss on drying, low water content or water activity excipient will help you to stabilize your formulation. Low nitrite grade excipients are used to control the nitrosamine impurity generation. Also, many of the excipient contain peroxides like povidone and copovidone are there. Then starch contain formaldehyde and microcrystalline cellulose like excipients contain the water quantity or they have higher loss on drying. So those things should be considered and controlled. Then API related consideration. First of all, you should know what type of API you are going to use into the formulation, whether it is crystalline or amorphous. Crystalline APIs have higher stability compared to the amorphous forms. API source grade route of synthesis, that is ROS, and impurity levels have the great impact onto the stability. So these are required to be assessed and controlled. Hygroscopicity, water content, water activity, loss on drying, pH of API solution, particle size of API and surface area of API. These all are the critical CMAs for stability. Also know whether coarser API can be used or you will require micronized API or fine particle size API. If your drug substance is insoluble, then many of the times the fine particle size API is used. Then understand the sensitivity of the API to specific pH, temperature or oxidation, sensitivity to light, moisture, hydrolysis, sensitivity to mechanical stress like compression, compaction and milling. Know the melting point of the API and correlate that melting point related information with the processing steps and processing temperature. Also know whether the API is volatile in nature or it is hygroscopic or deliquescent in nature. Understand and study the compactability of the API and compressibility of the API if you are working onto the tablet formulation. Solubility of the API, complex formation or adduct formation or solubilization has impact onto the stability. Compatibility of the API with the excipient plays a great role in the stability. Some APIs are available as API complex or API premix with the excipient. Sometimes the API itself contains some stabilizers like antioxidants. So all these parameters are required to be assessed and controlled. Then you can do the improvement for the stability of the formulation if you know the sensitivity of your API. Then process also has a great impact onto the stability. First of all, know your excipients, know your APIs, and then go for understanding the process related parameters for stability of the formulation. Use of water solvents in the processing steps or use in the process. If you know that your API is sensitive to water, it undergoes hydrolysis, then you should not go for using water or use a specific equipments like rapid mixer granulator. If the API is shear sensitive, use planetary mixer, that is PLM. Then use of HME should be avoided for those drug substances which are sensitive to temperature. For those drug substances, if you want to perform the solid dispersion, then go for spray drying instead of HME. So these are some basic principles. Go for direct compression instead of wet granulation or compaction. Wet granulation can be done by aqueous, non-aqueous or hydroalcoholic solvents. Dry granulation can be performed by slugging and roller compaction. Then temperature and duration of processing steps. In process checks, hold time and packs have the risk of stability. If you are holding the intermediate stage material for a longer duration time and if the pack used for whole time or holding the intermediate materials is not protective in nature, then it will give rise to increase in the impurities. Then in process controls like LOD and water content. If in process checks are not in place, if these are not controlled, then those things like LOD and water content of the in process steps may destabilize the formulation if the latter stages. Then use of FBD or FBP. Sometimes because of the oxidative nature of the API, the use of FBD and FBP is avoided. Some processes like pellet formulation require longer duration of use of these equipments. So for this, the API sensitivity to the process parameters is required to be understood. Processing environment conditions, that means the manufacturing conditions or manufacturing area environmental conditions like relative humidity. If you know that the product is sensitive to moisture, then go for low RH condition. You can use sodium vapor lamp if the API is sensitive to light. So it will require protection from light. If it is sensitive to oxygen, it will require protection from oxygen so that you can use nitrogen purging for processing or packaging operations. Use desiccant and oxygen scavengers, testing of input material and packaging materials. So many of the times it happens that the testing of input materials like API, excipients is insufficient. Same can happen with the packaging materials. And if you use those materials, then it can lead to stability failure. Then storage of the raw materials, APIs and in-process materials. If the storage is not proper, then you will end up with the product which is not stable. For example, if you use hard gelatin capsules for the formulation for manufacturing the batch, and if those capsules are not stored properly, then you will find that the moisture content of the capsule shells will change. It may become soft or it may become brittle due to the temperature variations and improper storage. Sometimes the gelatin shells may be contaminated with the microbial growth. Sometimes it is observed that due to improper storage of the gelatin capsules, cross-linking happens. So always have the risk assessment for these processing conditions, storage of the API and in-process materials and also the raw materials. Then use a planetary mixer if the API is sensitive to high shear so that you cannot use RMG. Study the effect of meal and meal type, meal speed and principles if API is shear sensitive. Processing time and conditions are required to be assessed. Packaging process and parameters are required to be evaluated. Melting due to heat generation during processing. Many of the time you will find that the API with low melting point will melt because of the processing steps like milling. Then cleaning of the equipment and processing area. Many times due to cross contamination, the impurity comes into the product and it may get increased due to interaction with the other components. Cleaning SOPs, cleaning process if breakdowns. Sometimes the equipment breakdown happens because of some reasons, and that uncleaned area is not, if it is not cleaned for the next processing steps because of the larger time gap, then there may be a chance of impurity generation or contamination. For example, the hot melt extrusion process If you break down the process for some time, then the material inside the hot melt extruder will remain in the equipment and it will be exposed to the higher temperature, which may lead to degradation of the API. And if that material is used in the next processing step, then you will find that the product is containing higher impurity. So these breakdowns should be controlled. If equipment breakdown is there, then cleaning should be performed for proceeding the batch manufacturing. Packaging related considerations. See, packaging is to protect the formulation from the environmental condition, from contamination, and it is also important for the transport. Also, for the use of the medicine by the patient or by the healthcare provider. For protecting the formulation from environmental factors, you can use stabilizers, desiccants, fillers. You can go for using silica gel or molecular sieve, oxygen scavengers. You can use fillers like cotton rayon coils if the tablets are friable. You can use charcoal if there is a bad odor generation into the formulation. Like you may have the experience of bad odor of some certain type of formulations like telmisartan, valsartan. So you can use charcoal that will absorb the bad odor. Then black lined packaging materials can be used for light sensitive APIs. You can use amber color bottles. You can use opaque blisters. You can use alu blisters or desiccant blisters. You can also use heavy weight SDP bottles and also stronger blister packs can be used. Then blister packs can be kept into the alu pouch to have better protection. Then co-packaging systems are used to separate the incompatible materials and also to pack the products which are taken together. Then for nitrosamine related impurities, avoid the use of nitrocellulose blisters. Then the main important thing to understand for stabilizing the formulation is that know the API, understand the properties of API, understand the requirement of the formulation, understand the in-use stability requirements if any, and perform the thorough risk assessment. And once you do the risk assessment, you can find out the ways to stabilize the formulation. Many of the formulations are there in the market which contain less stable API or unstable API, but due to formulation strategies and packaging strategies, those formulations are made stable. Also, if you find that the API and formulation both are sensitive to temperature, then you can do the stability studies, and based on those stability studies, you can change or you can recommend the storage conditions and the storage of the medicine in use. So I hope this video is basic for improving the stability of the pharmaceutical formulations and for performing the risk assessment. I will meet you soon with such kind of important and informative video, and in those videos we will understand about the each and every factor for risk assessment and for improving the stability of the pharmaceutical formulations. Thank you for watching the video. I request you to share this video with your colleagues and friends. Thank you.